This is Jake Fountain from the Department of Plant Pathology at Mississippi State University. In this poster presentation, we'll be talking about the identification of peanut late leaf spot resistance genes, specifically the pRPP13-like genes, using a nested association mapping approach. In this collaborative work, we utilized two nested association mapping populations, or NAM populations. The first was referred to as NAM TIFF-runner, the second is NAM Florida 07. These populations were constructed in a 2x4 NAM design, where you have two common parents, TIFF-runner and Florida 07 in this case, and four unique parents. And they are N08082, C7616, NC3033, and NBS-LRR R-genes, also known as SPT0606. These two NAM populations together totaled 1,076 recombinant inbred lines, with 580 in TIFF-runner and 496 in the Florida 07 NAM. These populations were originally developed by Dr. Coley Holbrook in Tifton, Georgia. These NAMs were planted in Tifton in 2015, 16, and 17, and were evaluated for a number of different traits, including disease resistance and agronomic performance. Here we're specifically talking about resistance to late leaf spot. These plants were rated on the Florida 1 to 10 scale at two different sampling points, either a mid-season rating, referred to in the poster as LLS1, or at harvest, referred to as LLS2. The NAMs were also genotyped using the newly developed 58K Axiom Arrakis SNP array, and linkage maps were then generated for these for QTL mapping. The raw genotypic data was then combined with the phenotype data for genome-wide association analysis as a second means of identifying significantly associated SNPs with late leaf spot resistance. For the linkage maps generated for each of the NAM populations, NAM TIFF-runner had 3,311 markers on its map, with a total length of 2,585.9 centimorgans. NAM Florida 07 had 2,668 markers on its map, with a total length of 2,393.4 centimorgans. These linkage maps, when combined with the phenotypic data, were used to identify 62 QTLs across both NAM populations, 30 in TIFF-runner and 32 in Florida 07. These QTLs ranged in phenotypic variance explained from 5.2% up to 65.6%, making these very useful as selectable markers in the future and useful for gene discovery. It is also worthy of note that examining major QTLs, that being QTLs with PVEs greater than 30% and being detected in at least 2 seasons, more were detected at the second leaf spot rating than the first in the NAM TIFF-runner population, but not so in Florida 07, where equal numbers were detected. This seems to suggest that the NAM TIFF-runner resistance could be more towards defoliation resistance rather than for leaf spots, as may be the case in Florida 07, given the functionality of the Florida 1 to 10 scale used for the ratings. For the GWAS results, NAM TIFF-runner had 221 significant SNPs, and NAM Florida 07, 293. Looking at overlap between the two sets of results, which is summarized in Table 1 of this poster, you can see quite a few areas where QTLs and the significant SNPs from the GWAS overlapped. In particular are those highlighted in green, which had SNPs associated with the pRPP13-like genes, or the Recognition of Peronospora Parasitica, which is a downy mildew. These genes had been previously associated with QTLs for other disease resistance in peanut, including Aspergillus flavus for aflatoxin. It's exciting to see the RPP13 type R genes being found at multiple locations, and seems to suggest that this family of R genes may be involved in late leaf spot resistance in peanut. For more questions or any concerns, please contact me at jcf416@msstate.edu. Thank you for your attention.